Well, Rich, apparently dinosaur soft tissue can last millions of years. Welcome to Revolution Against Evolution.
Absolutely.
I'm your host, Doug Sharp.
And I'm your co-host, Rich Gear, who's talking over him all the time.
That's right.
That's the way we roll. It's the first live show we've done like this where we're back to back, back in the studio together. It's pretty nice. Anyway.
So we're going to revisit this, and I think we've got our fingers deeply involved in this because I helped to donate the equipment at the Creation Research Society that they're going to be using to analyze the dinosaur DNA. Or DNA that looks like dinosaurs. They're doing other things like ostriches and alligators and
well, let's face it, dinosaurs are reptiles. Of course, the modern thing, Doug, is that the dinosaurs evolved into birds, which I think is basically horse feathers, but that's another issue. But the thing about it is that then of course crocodilians are reptiles, so. Those kinds of things kind of help out. I guess that really helps out because you want to find out, you want to dismiss all the objections to the things they're finding.
Right.
Okay? Because they're finding DNA, soft tissue, they're finding hemoglobin, red blood cells. They're finding, what else? They find
collagen.
Collagen, yes, soft,
biofilm presence, and so. They figure that if they find biofilm, soft tissues may survive in specific environmental conditions. So we have to figure out is what they are actually using to make the case that the dinosaur soft tissue can last that long. What they ended up doing, I think Mary Schweitzer is actually the one leading this.
She is an honest evolutionist.
Right.
To me, I mean, she is an evolutionary biologist, right? I mean,
you wouldn't think to look for soft tissue in dinosaur.
But she has an incredible curiosity. She's not out there trying to pontificate, puff. She's not really out there. She's actually kind of shy from what I was reading from some of the article. She's a little bit shy. But she's very curious. She's a brilliant person, but she's honest, and she's saying, well, wherever the science takes us, we're going to go there. And she's the first one that came up with red blood cells out of the T-Rex. Isn't it? Back in the mid-'90s, wasn't it? Mid-'90s, late 90s, something like that?
In the late 90s. And then in 2005 is when the research was published. And of course it took it took everything by storm. And that was 20 years ago, even 21 years ago. And so it's still sitting out there as a thorn in the side of the evolutionists, and the creationists are starting to pick up on it. They are organizing a fundraising campaign for this research.
Well, Doug, the thing about, let's go a couple things. Number one, why do you think it's taken so long for them to come out with this research? I have an answer, my own opinion of this. What do you think, Doug? Why it taking so long to come out with this?
Well, one of the problems is that if there's such an inculcation of this idea of millions of years that's been built up over the past 150 years.
Correct.
Where they talk about geologic time and deep time. And it's hard to get away from that if that's what you've been taught. And the other thing is that there is a implicit conclusion that they make that they would have to acknowledge a creator God, because the biblical timeline, which is what they set out to do.
To them.
Yeah, it is.
What they've done, I think one of the things, though, they've been doing, Doug, is trying to give, it's taken 20 years or 10 years or whatever, decades to come out with stuff, because they've been trying to figure out a way that, as you said in the beginning title, dinosaur DNA can last for millions of years. They're trying to figure out a way, a plausible explanation for how this stuff could last that long.
Right.
And I don't think they've done it. They've got a few plausible scenarios, or not plausible, but I think ad hoc explanations what they're using. To me, that's fascinating. To me, that's one of the reasons why it's taken so long for them to publish this stuff, is because they're trying to figure out ways to get around it. Because the obvious conclusion, Doug, is if millions of years do not exist, then you falsified the underpinning for Darwinian evolution. Because no one has ever seen evolution in a macrobiological sense at the genomic level. You have not seen any of this happen. My thought is that they've been trying to figure out a way to explain away the fact that soft tissue, DNA, and Doug, you even said that they've been finding carbon-14 in these things as well, correct?
Well, that's we can just suggest to the evolutionists some tests. Let's start out with carbon-14. I think there's.
All right, let's do that.
If you have carbon-14 in the sample, it would be something that would indicate as a young sample.
A young Earth, because the maximum age that they've been able to get out has been about 50,000 years or so. I mean, I think they extrapolate even out to 100,000. That's max. You're never going to get millions of years. And if you find carbon-14, then they have to explain it away with contamination or ability or mechanism for carbon-14 to. But it's pretty good when there are limits to what you can do, even at the most extreme grace period. If you want to give them, you can't get millions of years. You cannot get carbon-14 in a T-Rex or Triceratops or the Edmontosaurus. You're not going to get something in there. It should be nothing should exist after 1 million years, let alone 65, let alone 225 million, or however long they want to do the dinosaurs. So that's the one thing they have to come up with. So carbon-14. That to me, that's been a real bugaboo for many years. And it's interesting, Doug, when I was reading that article today, they were saying, well, creationists are coming up with, they're talking about, we would, because this would, on the face of it, Doug, falsify Darwinian evolution.
It would, yes.
It would falsify it.
The other suggested method that they could use on it is amino acid racemization.
What is that? How do you pronounce
racemization?
Racemization.
Yeah, remember I was talking about the right-handed and left-handed molecules in amino acids, and life only uses left-handed amino acids. Right. Yep. But if you have a sample that's dead, eventually that sample, because in the outside of the living environment, it'll naturally transpose into the right-handed kind.
Is that right? Okay.
So the racemization is where it decays, or it goes from left-handed to right-handed.
So it's kind of has a similar kind of idea that radiocarbon dating has, for different reasons, of course. But I understand that it's not quite. It's got quite a few more problems. Carbon-14, for all its issues, is much more standardized and well known than the, what did you call it? How's it?
Amino acid racemization.
Amino acid racemization. Okay.
And so racemization is a process of measuring, and it's the amino acid glutamine that is more prone to it than the rest of the amino acids.
But it's used more as a corroborative. It has to have something. Don't they use it like with carbon-14 or something else to kind of back up? Because I guess certain conditions can change the rate of decay of the amino acid, the racemization. Anyway, so yeah, those are two methods. And what have they? And Doug, there is one thing they did come up with. I don't know whether we want to get to that yet, but about hemoglobin or something, able to delay or slow down
that's
some of the process.
The explanation that they're currently working on is that somehow the iron in the hemoglobin is protecting the soft tissues from decaying. And how would they know that? You have to have some sort of mechanism to go back in time and
for their fossils, yeah. But haven't they done some testing to show in like modern day stuff that it does slow it a little bit, but not nearly what you need, not even close. It doesn't really. It's almost a moot point as far as its validity to show that millions of years. It can't even work for 1 million years, let alone tens to hundreds of millions of years. And we're finding stuff. What's interesting, Doug, all these things we're finding DNA, and we're finding soft tissue, collagen. We're finding hemoglobin. We're finding osteocyte.
I'll be want to clarify something here.
Okay.
I was with Joel Brown at the Creation Research Society, and they're looking for DNA.
Okay.
But they're not at the point where they can say
I thought they've actually found something.
it.
They've not found any. Okay.
But that is
I stand
something that
corrected.
they believe that there's a possibility of finding actually intact pieces of DNA in the samples. So if they do find that, it's another thing that DNA cannot last for millions of years.
No, they know that. None of this stuff. But I was looking at that one. You sent me Doug sent me a PowerPoint. I was looking at all the different kinds of soft tissue, the osteocytes, and there's all different kinds of things that they were finding and different kinds of things. And I go,
stretchy material.
Stretchy material is what it is. Yeah. For years, and what's the old expression? The old saying? I can't remember who came up with it, but your prejudice allows you what you can see. They never looked for any of this stuff, the evolutionists did, because there was no possible way that soft tissue, let alone red blood cells, or anything else could survive millions of years. So Mary Schweitzer, even though she was an evolutionist, as I say, an honest one, she started looking at stuff and found stuff. And of course, creationists have glommed onto that because we're going, if you're finding all this stuff, let's start looking for it. Yeah. And they're looking for it. Isn't it the iDino project? Isn't this something?
Yeah, that's what they're doing. Yeah. And so to go into a little bit more detail, what they did find, they tested demineralized bone fragments from 6 fossils, 4 T-Rex specimens, 1 Brachiosaurus, a Canadensis, and 1 Ceratopsian, which would be a Triceratops.
Well, like a it's a...
And Mark Armitage, he's got the Triceratops horn that he's using his electron microscope on.
And this side note there, he was let go from his career because he was coming out with this stuff.
Yeah.
Because he was basically revealing some of the implications of it, and they didn't like it. Mm-hmm. Now, Mary Schweitzer is not. She's kind of a professor emeritus, or she's pretty well respected because she's not backed off as far as evolutionary biology, but her honesty is refreshing in that in that framework. But he himself come out as he's a creations guy, and he got fired from his job, because of it. So there's a strong prejudice still in academia, in scientific circles, against any kind of biblical framework being used as a reference or substantiated by some of the findings they've been finding. Okay, back to what you were saying, Doug. So some of the stuff.
Well, Mark Armitage, he did manage to win a lawsuit where he regained his pension, but
not his job.
Not his job, but if you're starting to find something that conflicts with what the normal, or I would call it abnormal, paradigm is, then actually...
We'll say the dominant paradigm.
Yeah, right.
How's that? Which is evolutionary in long ages. There's no doubt about that. So that's the thing people don't understand is what is long... We've talked about this many times. It's always kind of good to revisit why long ages is so instrumental. Because it was really the first thing, Doug, almost at the very beginning, before Lyell even, the long ages with Hutton and some of those people, they gave an underpinning for long ages for Darwinian evolution to actually even happen. Because it's been an obvious thing, Doug, that no one has empirically seen something transition from one kind of an animal to another. Right. And yet, so they have to have long ages because no one has ever seen it. You can't demonstrate it at the genomic level. You can't remember that one thing was it Gillian Brown? Was it she asked Dawkins that question? Yeah. What's your best evidence of a genetic mutation increasing information at a genomic level? And he stumped him. It probably felt like 15 minutes, but it's probably more like 30 seconds or a minute, but it felt like forever on camera. And he said, I've been blindsided. Spent the next year trying to tell people how he was blindsided. It was a fair, legitimate question. And that's the point. No one has ever seen this. So they need long ages. Long ages was there before Darwin actually proposed his theory. Lyell was 1835. Hutton was 1795, I think, something like that. And Darwin was 1859. So I don't want to go into too much with the weeds, but those long ages is an underpinning for everything that allows for Darwinian evolutionary stuff to happen. All this folderol, and I like that old word, dinosaurs evolving into birds, all this kind of stuff. You can go in. They need long ages because no one has ever seen anything of the sort happening.
The only problem is that the long ages don't help. And the reason is if you're going to transition from one creature to another, and it doesn't take minute mutations. It takes a whole alteration of the environment in the organism and in the environment around it, And so, especially in the case of life from non-life, either
that's the big jump.
Happens.
How do you get there? Yeah.
Either it works or it doesn't. And if you have all this experimentation going on where you have mutations causing a change in the organism to create something else, usually that mutation is deleterious.
Yeah, usually it doesn't work very well, but yeah. Doug, sometimes I think the long ages is almost like a bait and switch environment, almost like the alien panspermia. If you haven't seen it, you can propose anything you want to. I told you one time I was at a youth group one time, and there was a young girl. She was probably 12, and she's kind of mature for her age. And I said to her, "Why is it that some of the best science fiction is always like evolutionary?" And she came out with the idea, "Well, they can make up anything they want." And I go, "Oh, I never thought. Oh, wait, I didn't mean it that way." Well, that's exactly what it is. You long agers can make up anything you want to because no one can ever test what you're doing. It's an article of faith. You tell me that I'm a faith monger, or faith operating only on faith, and I acknowledge the biblical text is my framework. But your Darwinian text or neo-Darwinian text is your framework, and so many things you appeal to, like the Big Bang, life from non-life, that has no empirical evidence whatsoever, an increase in genetic information to make it do for God, never been seen. So who has the more faith? I think you do. And the long ages is the underpinning for all of that. So finding stuff like that can't last for long ages, so cut to the chase in that sense, really undermines that whole scenario, I believe. Creationists have got, I would say, the trump card here. If you will, the thump card, because it to me, and that's why they spent 20 years trying to figure out a way that it could last for millions of years. And they still haven't figured it out. All their scenarios don't really work at all. Okay?
I wanted to go into how Mary Schweitzer did their experiments. They use transmitted light microscopy, scanning electronic microscopes, transmission electron microscopes, and nanocomputed tomography to capture extremely high resolution images of the vessels. And so
she's a brilliant lady, man.
Well, basically they just use various kinds of microscopes and a CAT scan.
Well, okay, sounds brilliant to me. But how they know how to read with those things? Anyway, but they also didn't they demineralize some of the bones to find some of the tissues and stuff.
Right. Yeah, that's what they ended up doing, and so they dissolved some of the bones in acid or something like that, and they got that isolated the soft tissues, and there they were, and so. They described it like this. It's like starting with a big funnel and working your way down, eliminating and identifying everything we see so that we can distinguish original structures and molecules from bacterial or fungal growth. And since we don't have a living control to compare the results to, we use ostriches. They say Edmontosaurus, the large duck-billed giant plant eater, lived along with Tyrannosaurus. And they also identified hydroxyproline, amino acid strongly associated with collagen in the bone. And so, according to this, represented an important confirmation that degraded collagen fragments were genuinely present inside the fossil.
Because that's their argument, Doug. A lot of times they think contamination, external things that got in there. That they're trying to eliminate all of that stuff. Okay,
and that's the caveat with research that the Creation Research Society is doing, is that if they find a sample and they find something unusual in that, they want to make sure it isn't something that's from contamination
Sure.
environment.
That makes
they
sense,
said
yeah.
this is probably a 5-year project to isolate any dinosaur DNA. And so it's you can probably tell that there's some work that went into Mary Schweitzer's paper before she ended up actually
Well,
publishing.
she knew it was going to raise a furor. It was going to, the maelstrom, the storm was going to be brewing, and but she's got enough credentials, and that she was able to weather some of that storm, so to speak. And she's still doing pretty well because, like I say, she's never recanted or come out as a creationist or by any stretch of the imagination. She may have some kind of a faith at some point. She's kind of real quiet about that in her own. She doesn't really talk about that. So we have no way of knowing one way or the other. But she's an evolutionary biologist, and she's never backed out of that, as far as I know.
Right.
And so her experiments have a lot of acceptance. See, that's a problem, Doug. Is if you come out as a creationist, you're automatically branded as you have no street cred. You have no recognition, which is unfortunate. But that's what happens when you end up with a dominant paradigm, Doug. The dominant paradigm has the ability to squelch debate, and they do it all the time. And it's happened for centuries, for millennia of time. The dominant paradigm, they don't like change. They don't like things they've been taught or have taught for years and preached and spread. Suddenly something comes out and says it's false. They have to figure out, okay, she's one of us, but we got to figure out, okay, how can we explain the long ages for the soft? It's because everyone knows. In fact, it was so codified into their theology. I call it theology. Is they never even looked for it, as we said earlier.
Right.
They never looked for it. They figured fossils were just mineralized bones. Mineralized. They weren't. Nothing was of the original tissue could possibly be there.
I was going to go back to amino acid racemization because I got some notes on it.
Oh, he does.
And
This is where the biology guy, the chemistry guy comes in. I got to tell you right now,
Okay.
and he's good at it.
And living organisms maintain amino acids almost entirely in the L configuration, which would be left-handed.
Left-handed. Okay.
And once the organism dies, biological control stops, and certain amino acids begins to racemize, which means they slowly convert from L to D until they approach equilibrium. The extent of this change is expressed as the DL ratio. D is dextro and L is levulo, which would be right and left-handed.
I wonder why they put a D instead of an R there.
Yeah.
Okay, all right.
So if you have a low DL sample ratio, it means that you have a relatively younger sample.
Okay.
If you have a high DL ratio, it's an older sample. And so if you have lots of the right-handed amino acid, it means that it's much older.
It's older. Okay. All right.
And so
But when we say older, how much are we talking about as far
we're
as
talking thousands of years.
thousands of years? Okay. Like carbon-14, only for
Yeah.
different reasons. Okay.
And different amino acids racemize at different rates.
Yeah.
And so this is relatively new. And so I think it's only been used on shells or bones or teeth or mollusks or eggshells or corals.
Yeah, that's what it's for. Yeah, it's for harder. Yeah.
Or sediments containing preserved organic remains. And so that's where this comes in. If we have preservation of organic remains, then it's used for, and it says it's useful for quaternary dating, which means recent dating. And so it's relatively helpful where radiocarbon dating is limited or unavailable. It can be used for relative dating with calibration, sometimes numerical age estimation. Now there's the problem. If you're calibrating it according to carbon-14 or other radioisotope dating methods, well, you have those dating methods which have that's their own suspect assumptions
exactly. Correct.
being used to calibrate amino acid racemization. So we have to be careful with this. And there's another problem is that it's sensitive to temperature, contamination or alteration, and different amino acids racemize at different times and rates. And then there's open system behavior in some samples, and so it's only something that you can mess around with and use that in cooperation with other.
Well, that's what I was talking about. It's usually a lot of times as a cooperation is not its own thing, because it's useful for corroborating a date that, let's say, carbon-14 gives you or something. It's very useful for that. So yeah.
So that's something I think is a suggestion that creationists and evolutionists should try to use on these samples if it shows that there's a high DL ratio. Well, it means it's old, and it's probably something that could be really millions of years.
Yeah, when you say old, we're not talking millions of years, we're talking thousands of years. So, you could get a date with carbon-14, 50,000 years, which we don't accept that either. But the point of it is, you're not dealing with 65 million. There's no way you're going to get that from these two methods of the. But Doug, that's the other thing. You said they're finding carbon-14 in some of these soft tissues as well. Are they finding? Did I understand you correctly when you said that to me?
I think that's correct. I can't corroborate that.
That's fine. We'll
I have to go
leave
back
that on
to
the shelf. Yeah.
But that's something that should be used to see if they corroborate that.
Absolutely.
Yeah. Well, I think that this is going to be a project that is going to need some funding. There is a website I'd like you to peruse, because they've set up a GoFundMe account for these projects, and this is one of many projects the Creation Research Society is starting to
Very good.
to do. We'll see you next time on Revolution Against Evolution.